Hello students and welcome to Home Learning Biology. My name is Mr. Cindzi, Mr. C. In today's presentation, we're looking at the alternative to practical paper as a continuation to what we were doing last time. So we'll start with question number C2, which reads, Figure 2.2 shows a fruit which has been kept in a dry environment for 1 day. Figure 2.3 shows a fruit which has been kept in a damp environment for 1 day. So here we are looking at two different environments. One is a damp environment, the other is a dry environment. And then we are comparing these two fruits with regards to a number of items out there, number of attributes. So here are the fruits we're talking about. Here is a figure 2.2 and figure 2.3. Kept in a dry environment for 1 day, that's figure 2.2. 2.3, kept in a damp environment for 1 day. Here is figure 2.2, as you can see here. That's 2.2. That's figure 2.3. And then we look at the question. Part 1 of the question reads, complete the table below to show one visible difference between the two dandelion fruits. So these are dandelion fruits. One visible difference. You can see that the one on the left, which is figure 2.3, is widespread like this at the top, and the one on the right is closed up, those things. What we call those features, as per the question, the features which are being compared here are the hair. So the feature is the hair. And for figure 2.2, the hair is widespread. So figure 2.2, the hair is widespread, which means it's spreading out, widespread. Whereas for figure 2.3, it is closed up. Closed up, that's the hair. Going back to the diagrams, going back to the two figures, the hair is right here. So that's the hair, which is widespread for this figure 2.2. And here it has closed up. Closed up. So with that, we have answered the question appropriately. 2.2, it's widespread, that's the hair. And 2.3, it has closed up. I want us now to look at the second part of the question, which now looks at how the closing up and the spreading out helps this plant in terms of this fruit, in terms of its adaptation. So the next part of the question reads, suggest how changing weather conditions would, 1, help disperse the fruits from the parent plant. Remember dispersal, fruit and seed dispersal. That's when the fruits and seeds are taken far away from the parent plant to avoid competition for sunlight, for water, for nutrients, for space. That is dispersal, fruit and seed dispersal. So here, what it was to suggest how these weather conditions, the dry weather conditions and the wet weather conditions, help in dispersing the fruits away from the parent plant. That's the first part of the question. 2, allow them to germinate in a new habitat. We've just said that fruit and seed dispersal help the seeds to germinate at distances far away from the parent plant to minimize competition for the water, space, sunlight, and the like. Now, let's have the two fruits in the picture. We said the first fruit was placed in a dry environment. That is why the hair was spread out. And the second fruit was in a damp environment. So we've got two weather conditions here, dry and damp. So our answer will be based on these two weather conditions, dry and damp. Let's start with the dry. This question here is worth how many marks? Let's see. It's worth 5 marks. So we should at least mention 6 points, or rather 5 points, in order to put on the right track. So let's talk about the dryness, the dry fruit first. If the fruit is dry, the hair is dry, So the surface area is increased. So the dryness will increase surface area. The dryness will increase dry. So this one will increase surface area. It's spreading out, right? It has spread out. So there's more surface area, increased surface area, increased surface area. If the surface area is increased, then it will easily be carried away by the wind. So easily carried away by wind. By wind. That is the dispersal easily carried away by wind, which is the wind dispersal. Easily carried away by wind because it is dry. And then it will land distances away from the parent plant. Land distances away, distance away from parent, P for parent, plant for plant. P for parent, the other P for plant. So it's dry. It will increase surface area for it to be easily blown away by the wind. Then it will land distances away from the parent plant and avoid competition. Avoid competition or minimize competition. Avoid competition. I like minimize though. Minimize competition for sunlight, space, nutrients, water. You list them out. So that's with regard to the dry fruit. Now let's get to the wet fruit. The wet fruit, its advantage is that it's wet. It's got moisture. The moisture content is a requirement for the germination. The moisture is a requirement for the germination. Germination. The moisture content is a requirement for the germination. Remember, for germination to occur, there must be moisture, there must be suitable temperature, there must be suitable temperature, moisture, and oxygen, right? So the moisture is a requirement for germination. What does the moisture do? What exactly is the contribution of the moisture there? It will activate enzymes. So let's write that one down. Moisture will activate enzymes. Activate enzymes. Activate enzymes. So we now have a number of points here. Let's count the points. We were required to mention at least 5 points. How many do we have? 1, 2, 3, 4, 5, 6. We get all the points. There are 5 points which are awarded to us. Let's say this in summary again. Looking at the question says, suggest and explain how changing weather conditions would help disperse the fruits away from the parent plant. We start with the dry weather condition. It's dry. So the hair offers an increased surface area because it's widespread. The hair offers an increased surface area because it's widespread. If it's widespread, then it offers some kind of resistance to the air. Then the air will blow the fruit away because the surface area is offering some resistance. Easily carried by the wind, that's point number 2, due to the large surface area. And then it will land away, distances away from the parent plant, where it will then germinate and minimize competition for water, nutrients, light, space. So mention all those 4 points. And now getting to the dry fruit. The dry fruit has high, or rather wet fruit. The wet fruit has a high moisture content. Moisture, that is water. Water, which is a requirement for seed germination. In what way? It will activate the enzymes, which will thus lead to the seed germinating. So, having said that, we are sure of getting all the 5 points. We mentioned about 7 points here. So, yeah, that's how this question here is answered. Let's move on to the next question, which is question number C3. So, which reads, Figure 1.1. Figure 3.1 shows sections through a ginger and lotus stems. So we are now looking at stems. We're no longer looking at the fruits, but we're looking at stems now. So figure 3.1 shows sections through ginger, that's one. Zingiber officinaire, that's the biological name. And the lotus, Nelumbo nucifera, that's the biological name again. The species on the right, and the genus here. The genus and the species. That's the way the name is given. So this figure 3.1 is showing two different stems of plants. The ginger is on the left, as you can see. That's the ginger here, and that's the lotus. Let's look at the questions. A1, state one visible similarity between the two stems. One visible similarity between the two stems. Let's look at the two stems closely. These stems have got a bark on the outside. The bark is always on the outside, so there must be a bark. So one visible similarity, they both have a bark. The bark is this part here. That's the bark. And this one here, that's the bark. They both have a bark, that's a similarity. Part 2 of the question reads, complete table 3.1 to show 3 visible differences between the two stems. The keyword here is visible. These differences must be visible. Don't think about a stem we have seen somewhere, then you say, oh, a stem that is like that has such features. So we're looking at visible as per this slide, as per this figure, the figure given here. Visible differences. Let us look at them. Let's start with the bark. They both have a bark, but the bark is different. The bark for the ginger on the left is rough. The bark for the lotus on the right is smooth. That's with the bark. Let's get inside the stems. The ginger on the left has got fibers. The lotus hasn't got the fiber material there. Fiber, that's the fiber. This part here, that's the fiber. There is none on the right for the lotus. That's difference number 2. Difference number 3. On the left, the ginger has an irregular outline. That's the outside. It's irregular in shape. Whereas the lotus is roughly circular. It's more circular. It's more circular in net. Look at it this way. It's more circular. This one is more irregular. That's the outline. That's another visible difference. That's difference number 3. Difference number 4. The lotus has got holes. The holes right in the middle. You can see the holes here. These are the holes. Whereas ginger hasn't got the holes. So I've mentioned already 4 differences. Then the color. You can also talk about the color. The lotus has got some black patches. That's inside, just around the hole, some black patches in there. Whereas the ginger, the color is more uniform. So that's 5. We've talked about 5. We mentioned 5 differences. Let's see how many the examiner needed. 3. So out of 5, we have talked about 3. Let's write them down. We say the ginger has a rough outline, rough outline, whereas the lotus is smooth outline. That's with regard to the bark on the outside. Then for the lotus, you talk about holes. There are holes here. For the ginger, no holes. Didn't see holes there. None. Let's say holes, no holes, or none. Let's say no holes. For the ginger, you talk about fiber material, fibers material, no fibers, yeah. Then you also talk about the color. You say the lotus has a variety of some dark patches, whereas the ginger hasn't got that. Let's go back to confirm our differences. The ginger on the left and the lotus on the right. Those are the differences. Having said that, having written all that, we are sure to attain to get all the marks awarded to this particular question. Now let's go to the next part. of the same question, which is question number C B1. Lotus plants live in water. Lotus plants live in water, okay. Lotus plants live in water, as you have seen them. They've got holes. We saw the holes for the lotus plants. And we're being told now that they live in water. Lotus plants live in water. Let's underline the water. Suggest and explain an adaptation of the lotus stem in the water habitat. Let's look at the marks. 2 marks for that. Suggest and explain. So we are suggest, give a point, then explain. An adaptation. We define adaptation as a feature that the organism will develop in order to survive, in order for it to survive. So we're saying here, suggest and explain an adaptation of the lotus stem in the water habitat. An adaptation of the lotus stem in the water habitat. Suggest and explain adaptation of the water stem in the water habitat. So the lotus has got holes. That is an adaptation. What's the role of the holes in the stem of the lotus? They keep the lotus plant upright. They make it to float. They make it to float in the water. Flotation. That's an adaptation. The lotus stem has got holes as seen from its cross section, and these make the lotus plants to float in the water. To float in the water, that's flotation. Part C, the cells of lotus contain starch grains. The cells of lotus roots contain starch grains. Let's underline starch. Describe how we'll prepare a microscope slide of cells of a lotus stem to show starch grains. So what this question requires of us is a test for starch. Preparation of your specimen as you are going to test your specimen for starch. 1. How would you test for starch from a lotus stem? You cut it into pieces. Cut it into small pieces. Cut into small pieces. You may even grind it. Grind it. Pieces. You may even grind the stem into some bit of powder. Grind it. You may even grind it. Cut into small pieces or grind. That's number 1. Number 2, you place on a white tile or slide. Let's say slide. Place on a slide. The cut pieces are placed on a slide. Number 3, point number 3, you cut it, you place it on a slide. Point number 3, we're testing for starch, remember? Point number 3, you add iodine solution. Add a few drops of iodine solution. A few drops of iodine solution. Point number 4, cover slip. You cover with a cover slip. Cover slip on top. You cover with a cover slip. And then after that, you observe the color change. If the color changes to blue-black, it means starch is present. Blue-black color means starch is present. So these are the steps. It's 4 marks. We've mentioned 5 points. Let's go through the points again. Starting with the question. Describe how we prepare a microscope slide of the cells of a lotus stem to show the starch grains. Point number 1, you cut the stem into small pieces, very small pieces, cut them, or you grind them. 2, place them on a slide. Point number 3, add the iodine solution. Step 4, place a cover slip. Step 5, if the color changes blue black, it means starch is present. So these are the steps you go about when you're testing for starch in any plant material, but with reference to the question in the lotus stem that we are looking at right now. Let us go to the next part of the question, which is question number 1 from a different paper, still looking at the alternative practical paper. Some students compared the metabolism of 2 yeast mixtures in test tubes 1, W1 and test tube W2 using the apparatus shown in figure 1.1. So we are comparing here in this activity, we're comparing the effect of yeast mixtures, underline yeast mixtures, in test tubes W1 and test tube W2 using the apparatus shown in figure 1.1. Both mixtures contain the same concentration of sucrose. What are we saying here? There is sucrose in here. That's W2. There is sucrose, a sugar, and there is also sucrose here. And then yeast is added. Yeast is added, okay, plus yeast, then plus yeast. Yeast is added in both test tubes. There is a delivery tube going to the right, and on the right, there is water. There is water here. The delivery tube is going to the right. There is water. We can see bubbles here. These are bubbles. It shows that a reaction is taking place in the test tubes. Look at the temperature. The temperature of the water is at 30 degrees Celsius to 40 degrees Celsius. And there is a thermometer. Let's look at the diagram again. Test tube W2 on the left, test tube W1 on the right. In both test tubes, there is sucrose, a sugar. In both test tubes, yeast mixtures are added. 20 cubic centimeters active yeast mixture in the test tube added there. And then, as the yeast is added into the sucrose, the delivery tube will take a gas which will be bubbled in the water on the right. As you can see, the bubbles. Of note here is that the students conducting this activity, practical activity, are starting with W1. They will then do W2. Within the same test tube, whose temperature is 30 degrees Celsius to 40 degrees Celsius initially. Note again that the temperature here is not regulated. There's nothing heating up the water here. There is no water bath. Remember, water bath has a constant heating process. So initially, when the temperature was 30, let's say it was 40, then as the activity progressed, chances are the temperature will drop. It was 40 for W1. That's an assumption, 40 for W1. But when it's time for W2 to be investigated, the temperature will definitely be slightly lower. So we will be looking at the questions with regards to this drawing here. We'll be looking at loopholes with regards to conducting this particular activity. So let's look at the figure again for the last time now. There's a thermometer, W2 already connected to the delivery tube, to a test tube on the right, with the bubbles of the gas collected. That's W1. W2 is still in the line. After time X, where observations have been made for W1, then the students will then do W2. And the yeast, the active yeast mixture in the test tube, Yeast already added there. We don't know how much yeast was added. But both mixtures contain the same concentration of sucrose that we are told. But with regards to the yeast, we are not sure. We are not being told that equal amount of yeast was added. Now it's time for the questions. The apparatus was left for 2 minutes. Let's underline 2 minutes there. The apparatus was left for 2 minutes. After this period, the number of gas bubbles released from the delivery tube was counted for 2 minutes. Counted for 2 minutes. This number was recorded as trial 1. That's table 1.1 as you're going to see below. The yeast mixture was shaken. Yeast mixture was shaken. And the number of bubbles was recorded. Number of bubbles was recorded for 2 more minutes as trial 2. This was repeated for trial 3. The yeast mixture was shaken. You shake. Like you have a test tube, then you shake it like that. You shake it like that. Remember, in a laboratory, you can use equipment, sophisticated, advanced equipment for stirring. Shaking, literally shaking yourself as a student. You may not shake as one would expect the mixture to be agitated. You may shake one test tube differently from the other test tube. So probably that's another loophole, but let's go further and look at the question. The whole procedure was then repeated using test tube W2. So from W1, the students, having finished W1, they went to W2. W1, remember, is test tube 1. Taking you back to the drawing, to the diagram, the figure. W1 is that test tube, and W2 is the other test tube there. So it was there, once it was done with W1, the students then went to W2. No heating up of the water, so the water was bound to cool a bit in the process. The results for all the 3 trials for test tube W2 were recorded in table 1.1. Let us look at the results now. Yeast mixture, W1 and W2. These are the yeast mixtures, W1 and W2. Trial 1, trial 2, trial 3. The trials means doing the activity almost exactly like you did prior. Like trial 1. In trial 1, the yeast was added to the sucrose, then the bubbles were observed and counted. And the count was 5. Trial 2, still with W1 test tube, they were counted after 2 minutes, 3. Trial 3, after another 2 minutes, 2 bubbles were recorded. The same 2 minutes which was underlined here. The same 2 minutes which was underlined here. That's the 2 minutes. That's the time for, the waiting time before the observation was 2 minutes. And that's W1. W2 is test tube 2. The number of bubbles observed for test tube 2, the one which had been in the waiting list for W1 to finish, then W2 into the water. The number of bubbles observed were 20 initially. 20 bubbles. The next 2 minutes, that's trial number 2. Second trial, 15. The third trial, 10 bubbles. Now it's time to look at the questions. Question number 1 reads, gas bubbles are produced in this experiment. Yes, gas bubbles are produced in the experiment, as you can see from the table here. The gas bubbles are produced. State which metabolic process is being carried out by the yeast cells to produce this gas. Metabolic process. Let's go back to our figure and see what exactly is happening there. We have yeast. We have yeast here. Yeast on sucrose. Yeast on the sucrose, the sugar. So we've got yeast on the sugar, yeast on the sucrose. What's likely to happen there? Fermentation reminds one of fermentation, the brewing industry. yeast, baking industry, the baking processes, baking and brewing. Yeast added on the raw material, the carbohydrate, will produce carbon dioxide, will produce alcohol, will produce all that. So there is respiration and fermentation. And respiration produces which gas? Carbon dioxide gas. Fermentation produces what? Alcohol and carbon dioxide gas. So the process here, if we give as one process fermentation, we are on the right track. We're correct. Fermentation. If we say again, the process is respiration. Respiration, we are also correct. So the processes taking place there are fermentation. Is there fermentation, respiration? If you have fermentation, respiration, that's okay. Name this gas produced. We've just said that the gas produced is carbon dioxide. Carbon dioxide is the product of fermentation, the product of respiration. So with that, students, I want us to take a short break. We'll come back and look at part B of the question. So stay with me. I can't sit. I can't sit. Welcome back, students. Let's look at part B of the question. The question on fermentation, respiration, where we're looking at yeast cells in the sucrose solutions conducted by the students in their chemistry, in the biology laboratory. Part B says, suggest why the test tubes W1 and W2 were placed in a beaker of warm water during the experiment. That is 2 marks. Were placed in a beaker of warm water. Warmth. Remember when you talked about conditions necessary for germination, we said warmth. So warmth will increase the reaction. So the warm condition will increase the reaction. The reaction, Rn for reaction. The reaction of the yeast to the sucrose. To make it happen faster, we increase it. Also, the warm condition will serve as a control. That's a control temperature. That's control temperature. Control temp. It will be improper to have WA in a cold condition and W1 in a cold water condition and W2 in warm water. They must be the same temperature, almost the same temperature. That is a control. The control is saying the conditions must be the same. So that's a control temperature. If you've got control to serve as a control, that's correct. If you have increased the reaction, that is correct. Part C reads, describe and explain differences observed in the number of bubbles of the gas released. Describe and explain the differences observed in the number of bubbles observed. For this question, we need to go back and look at our table with regards to the number of bubbles. For W1, we can see that for trial 1, the number of bubbles is low. Trial 2 is lower. Trial 3, it goes down. So for all test tubes, W1 and W2, trial 1 records a high number of bubbles. Trial 2, it goes down. Trial 3, it's even lower. But W2 is higher, has a high number of bubbles than W1. So the question says now, we have to account for those differences. What can we say about it? What makes those differences? Let's look at the question again to be specific, to be exact on what the question needs. Suggest why test tube W1 and W2 were placed in the beaker of warm water during the experiment. We've done that. C says, describe and explain any differences observed in the number of bubbles of the gases released. Number of bubbles of the gases released. W2, let's say W2. Let's start with W2. That one is 3 marks. With W2, chances are there were high bubbles, all right? A high number of bubbles. More bubbles here. More bubbles. Then W1, less bubbles. There's B. Why? If there are more bubbles in W2, chances are there was more yeast added. More yeast added. Remember, we said the yeast was not quantified. The yeast added in the test tube was not quantified. So chances are there was more yeast added. There was more yeast added. In W1, there was less yeast. Less yeast with W1. What else will account for the more bubbles in W2? The time, the exposure time. The students are not carrying a watch, a clock, a timing device. That's not mentioned. So chances are W2 had more exposure. The yeast in W2 had more exposure to the sucrose. More time. So that's more time for the reaction. More time for the reaction. That is why more bubbles. Whereas here, less time. Chances are there was less time. That is a suggestion, remember? Describe and explain. Describe and explain the observations. Describe and explain any differences observed in the number of bubbles of the gas released. So we've already mentioned 4 points, and 3 points were made here. Differences observed in the number of bubbles increase. Let's talk about the trend. Look at W1. We said there was 15, trial 1. Trial 2, 10. Trial 3, 5. How do you account for the decrease in the number of bubbles as a difference between trial 1, trial 2, trial 3? Those are different. Those are the differences you have to account for. We've got to explain. How do you explain those differences? Trial 1, because you had more bubbles, chances are there was still a lot of sucrose in the trial 1. I'll write T1 for trial 1, and then more sucrose. More S for sucrose. In trial 3, all right, less sucrose. Less sucrose. The next difference, students, is with regard to the trials. Remember, for test tube W2, 3 trials were conducted. Trial 1, trial 2, and trial 3. Likewise, with test tube W1, 3 trials were conducted. And moving from trial 1, let's take W2 for instance. W2 had 15 bubbles, trial 1. Trial 2, 10 bubbles. Trial 3, 5 bubbles. So the trend is a decrease in the number of bubbles as per the trials. What will cause this? In the first trial, chances are there was a lot of the sucrose, sufficient sucrose. Going to trial 2, most of the sucrose had already reacted with the yeast, and as such, the number of bubbles decreased to 10. Trial 3, even more sucrose had reacted with the yeast, and so the number of bubbles decreased. So with that, we can say the trials were as a result of, the differences were as a result of the sucrose reacted down the trials. Likewise here, the sucrose reaction with the yeast resulting to a reduced number of bubbles at trial number 3 compared to trial number 1 for both cases. With that, students, this question has been answered correctly, appropriately. We've given 1, 2, 3, 4, 5, 6 points out of a maximum of 3 allocated for this question, which means you've done very well with this. Let's go to the next part of the question, which is question number page 4, which is question number, part D of the question, which reads, state 2 sources of error in the method of this investigation and suggest how to improve this method to reduce each source of error. So we're looking at sources of errors with regards to how the students are conducting this investigation and ways of improvement, how it could be improved. Sources of error, number 1, the temperature. Sources of error, the temperature. Temperature, lack of temperature control, lack of temperature control. Lack of temperature control, a regulator. We're told that the temperature was between 30 to 40 degrees Celsius. Leaving the water right there under the environmental conditions, like the temperature, room temperature, chances were the water will get cooler from trial 1 to trial 3. By the time trial 3 was done, the water will no longer be at the temperature it was initially. So that is an error. There's an error. There's a source of error. How can it be improved? Having a water bath. A water bath as means of keeping the temperature constant. Water bath as means of keeping the temperature constant, or constantly heating up the water and having a thermometer to measure the temperature to ensure that as you conduct trial 1, trial 2, trial 3, the temperature is maintained at that level. So that's an error with regards to temperature. Source of error number 2, the shaking. We're told that the test tubes were shaken. Shaking of the test tubes by hand, probably by hand, because these were students. They were shaking the test tube by hand. We know that shaking will facilitate the rate of the reaction. It's like stirring. So the rate, the force, the magnitude of shaking will differ because these are students again. Test tube A will be shaken a little gently, and test tube trial 3, probably they'll give a harder shaking. So the rate of agitation will be different. So that's the shaking. So there must be a steering mechanism as an improvement, a steering mechanism, so that it's uniform all over. Steering mechanism for shaking, for facilitating the rate of the reaction. That's on the regard of the shaking. Other sources of error, the timing. We're not being told that the students were having a stop clock for timing the 2 minutes. So that also should be provided and should be well done for such comparison, purposes between trial 1, trial 2, trial 3, and also between W1 test tube and W2 test tube. So these are the errors and many other errors out there which may be, improved, may be rectified in this regard. I want us to look at the next part of the question, which is question number 3. This one is slightly different. We have now moved away from this activity of the students. Figure 2.1 shows an upper surface of 2 leaves. We're now looking at leaves W3 and W4. Still, this is the alternative to practical paper. W3 and W4, that's the upper surface of the leaf. This W3 and that's W4. Make a large labeled drawing of the leaf W3. That's part 1, that's part A, 2A. You're making a large drawing which you must label a drawing of W3. Let's quickly make it here. We'll make it large, W3. There is this part here, then it goes like that. That's the middle. That's a rough sketch of the leaf, right? It's large. Compared to that one, it's large. Then labeled. That's the apex here. The tip will be the apex. That's the midrib. That's the petiole or the blade, or the leaf blade. Then you've got veins going like that. Then you have veins here going like that. Those are the leaf veins. These are the leaf veins going like that. deep inside. These are the veins. And that's the petiole. That's the enlarged leaf, drawn and labeled leaf W3. It will look something like this, but you can do it better there. Let's look at the next part of the question. But before I go there, because in the next part of question 2, that's 2b, we'll be comparing, we'll be looking at the differences between W3 and W4. Let's look at them before we get to the actual question. Look at the sides of the leaf. It's like serrated in both of them. Serrated. And W4 has leaflets. It's a compound leaf. It's got leaflets. It's a leaf with small leaves. That's called a compound leaf. Whereas W3 hasn't got the leaflets. It's a simple leaf. Hasn't got the leaflets. Both leaves have got the veins. And the midrib is there. Look at the tip. The tip for both leaves. This one is sharp. A sharp tip. This one is more rounded. Serrated. Serrated here. Serrated. Both are serrated. Leaflets here, no leaflets here. With that, let's get to the second part of the question, where we'll be looking at the differences between these two leaves that we've just seen in this question of the alternative to practical paper. Carefully observe leaf W3. We have already done that. And W4 in figure 2.1. We have done that. Describe one similarity. Let's take one similarity and two differences that you can see. Do not include size in your comparison. A similarity, both leaves have veins. We saw the veins. We saw the veins in both leaves. And both leaves have the midrib. Midrib. They said one similarity, but the veins are there. The midrib was there. We saw it. Let's go to the differences. Two differences. W1, which was the simple leaf, had a sharp apex. Sharp apex. Remember, we even labeled that sharp apex or sharp tip. Whereas W2, it's blunt or rounded. Let's say rounded. Rounded apex. Rounded tip. Rounded tip for W2. W1, another difference with regards to W1. It's a simple leaf. W1 is simple leaf. Simple leaf here. Whereas W2, a compound leaf with leaflets, right? If you write leaflets, you're on the right track. That's correct. Or write compound leaf, compound leaf. So we've given the differences. Sharp apex, simple leaf, W1, W2, rounded tip, and the leaflets are there. The serration, that's a similarity because both leaves were serrated at the sides. So you can write that one here. Both leaves are serrated at the sides, you know. With that, let us go to the next part of the question, which is question number 4. It's a different question. We're now looking at the cross-section of the leaf. That's one of the leaves. Probably the simple leaf, the cross-section of the simple leaf. Let's look at this one here. Figure 2.2 shows a photomicrograph of a section of a leaf to W3. Yes, W3. It's a cross-section. It shows a photomicrograph of a section of the leaf. That's W3. The simple leaf. Cross-section. That's how it looks like. Look at the cell Y and AB is labeled there. For information, remember the palisade layers here, palisade cells. Remember the spongy mesophyll cells. Remember the stoma. That's the stoma enclosed by the gut cells. The air spaces here. Air spaces. The lower epidermis. All right. Lower epidermis. That's the lower epidermis. And then here it's the upper epidermis, UP, UE, that's the upper epidermis. The cuticle here, all right, cut or cuticle. Palisade, we said this is palisade palisade layer. The spongy mesophyll cells, And the palisade cells, we know that they are rich in chloroplasts. The chloroplast right here for photosynthesis. Chloroplast right here for photosynthesis. As a briefing of background information with regards to the cross-section of the leaf. With that, let's get to the question now. Figure 2.2. On figure 2.2, draw a line to label a photosynthetic cell in the palisade layer. Wow, we've already covered that. We said that's the palisade layer. Draw a line to show the photosynthetic area. So that's where we place the chlorophyll. We denoted the chlorophyll. So that's the line. Photosynthetic area. So in this question, we are being asked to identify the palisade layer because in a leaf, most photosynthesis occurs in the palisade layer where they are the chloroplasts. Remember, chloroplasts contain the green pigment chlorophyll which traps the sunlight for the reaction in there. So that's where we are. That's the line. Draw the line, then you draw the line as we have done. Photosynthesis. That's where photosynthesis occurs. Let's look at the question to be more precise. Draw a line on figure 2.2 to show the pathway that, the first part. On figure 2.2, draw a line to label the photosynthetic cell in the palisade layer. We have just done that. That's the photosynthetic cell right here. We've drawn it. The second part of the question reads, draw arrows in figure 2.2 to show the pathway that carbon dioxide gas must take to reach the photosynthetic cell labeled C1 from the outside of the leaf, from outside the leaf. So what I'm saying here, we're drawing a route that will be taken by carbon dioxide. A route that will be taken by carbon dioxide as it moves from the atmosphere into that photosynthetic area we highlighted. So this is the route. This is the route. First, we start from the A, from the atmosphere. Here we are. That is the A here. Then we get in through the stoma. Because we've labeled the stoma this side, let's use this route. So this is it here. That's the route. It goes in through the stoma, goes like that. We're going to the photosynthetic cell. Okay? It moves through the air spaces. All the way up until it gets to that dot. Let's give it a circle. That's the photosynthetic area. Remember, that is similar to this one here. So these are both the photosynthetic areas, which we labeled in part 1 of the question. So, following our route, right from the atmosphere, let's start all over again. So that is the carbon dioxide, CO2 gas from the atmosphere, going like that, getting in through the stoma, the air spaces, spongy mesophyll cells moving in between, moving there, right there, and then moving up until getting into the palisade cells and into the chloroplast and chlorophyll. Once in the chloroplast, it will find the chlorophyll, which has absorbed the light. Then there will be water there, the carbon dioxide, water, and chlorophyll. Photosynthesis will occur in the palisade cell. So with that, let's look at the marks awarded to this part of the question. That is 2 marks. This part of the question is 2 marks. So having drawn that, we get all our marks, all the 2 marks. Let us now go to the next part of the question, which is question number, part 6 of the question. That's D, part D. Measure the length from A to B of cell Y on figure 2.2. Record your measurements, length A, B. Record your measurement, length A from B. Calculate the actual length of cell Y. Show your working. Let us say we measure the distance, the AB distance, the AB length. They want us here to calculate the actual length. We'll go to that slide. We're to measure AB, then calculate the actual length. Not the length that is we shall have measured there. We'll calculate the actual length. Let's go back to that slide. This involves a bit of mathematics. So let's get here. This is where we are. We are requested to measure the length of the cell Y, AB of cell Y. And this is approximately 2 centimeters. We have to measure those. Let's say it's 2 centimeters. 2 centimeters, the length is 2 centimeters. That's the length from this figure. But they want the actual length. This diagram here, this photomicrograph has been magnified. That's an important aspect of it. Has been magnified. We have to look at the magnification. Magnification is 250. Magnified 280 times. Which means the actual length will be the 2 centimeters divided by a right act length. Act length equals 2 centimeters divided by the magnification, which is 280. 0.007. The answer is 0.007. So the key point here is to consider the magnification. The actual length as measured from the paper is 2 centimeters. Once they say actual, then it's going to be the 2 centimeters divided by the magnification, which here is 280. Remember, we're looking at a plant here, a cross-section of a plant, a cross-section of a leaf, from one point to another point. So the length can definitely not be 2 centimeters. It has to be very, very, very small, and 0.007 centimeters does make sense. So students, that marks the end of our revision with regards to the alternative to practical paper. You do need to prepare yourself very thoroughly for this paper. And if you'll take the practical paper, you do have to acquaint yourself with all the practicals you've been doing in the classroom for the science laboratory, biology laboratory, for definitely to be like a reviewing of all those things you've done. So from me, Mr. Cindzi, Mr. C, I'm saying have a very, very lovely evening. Enjoy the rest of the programs on Swazi TV. Bye.